We are now at Chapter 15, Microbial Mechanisms of Pathogenicity. Let's talk about some terminology. Pathogenicity is the ability to cause disease. It's the ability of the microbes to cause disease. It's the ability of the microbes to cause harm. Virulence is the extent of pathogenicity. How much harm, the degree of harm, how much, like on a 0 to 10 scale. Do you have the sniffles, a little bit of a runny nose, or are you bleeding out of your eyes? How much harm is happening? The extent of the pathogenicity. Is it mild? Is it survivable? Is it likely terminal? That's what virulence is. How much harm, the extent of pathogenicity. For upcoming quizzes and exams, you need to know the periods of disease. You need to know the name of each period, and you need to know what is happening in each period. The first period of disease is incubation. It's after the initial entry of the pathogen. It has entered the body somehow, and the pathogens are beginning to multiply. You are not having signs and symptoms at this point. Things are just beginning. The bacteria is beginning to multiply. The virus is beginning to multiply. Then comes the prodromal stage. In the prodromal period, pathogens continue to multiply. They're still multiplying, but this is when signs and symptoms just begin. You might have a scratchy throat. They're very mild. Signs and symptoms in the prodromal stage are mild, but they are present. In the period of illness, signs and symptoms are obvious and they are now severe. This is when you are sick. It is very obvious that you have an infection happening. Then you have the period of decline. The pathogens and the signs and the symptoms, they all begin to decrease. You're not as sick as you were in the period of illness. You still are sick, but things are starting to get better. Then you have the period of convalescence. That's when you're taking the time to regain your strength, to get all better. Sometimes the period of convalescence, depending on what the infection was or what the disease is, can take a very long time. If it's a very serious illness that you have gone through, the period of convalescence may take quite a while for you to get back to being 100%. This is an excellent representation of the periods of disease. The progression of an infectious disease is divided again into those 5 periods that you need to know. If you look at this, you can see that the 5 periods are related to the number of pathogen particles in red and the severity of the signs and symptoms in blue. I really like this representation because you can see how they correlate with each other. Remember that incubation period, you don't have any signs and symptoms. That's when the pathogen has entered the body and it begins to multiply. That bacteria or that virus, that fungus, whatever it is beginning to multiply in that incubation period. Then you have the prodromal period. That period is when the signs and symptoms start, but they're very mild. You have a scratchy throat. Your nose might be a little runny. Then the pathogens are continuing to multiply, and you can also see that the signs and symptoms are increasing throughout the period of illness. That is when you are really sick. That is when the signs and symptoms are obvious and the pathogens are at their peak. Then you crest over that hill and you go into the period of decline. That's when the pathogens are decreasing. That is when your signs and symptoms are decreasing. You're still sick, but you're getting a little bit better. Then you're going to enter that last period of disease, the period of convalescence. That's when you're getting better. That's when you are going to get back to 100%. But sometimes it takes a really long time to gain your strength back after having a very serious infection. Sometimes the convalescence period will be shorter. It really depends on the illness or the infection. Portals of entry are how those pathogens get inside, how those microbes get into your body to cause infection or diseases. Mucous membranes line your digestive tract, your respiratory tract. They are in your oral cavity, your conjunctiva of your eyes, your urogenital tract. Pathogens can get into these areas, and oftentimes the mucous membranes can help the immune system by trapping these microbes. They're wet, they're sticky, they can trap the microbes, and sometimes they can neutralize the microbes. But if a pathogen can get past the mucous membranes, then a disease or an infection can result. You can breathe in pathogens into your mouth that go down into your respiratory tract. You can swallow pathogens. When you swallow pathogens in your food, that's how it can get to your mucous membranes of your digestive tract. You also have the skin. It's an excellent barrier, but it's also a portal of entry. Microbes can get in through hair follicles or sweat gland ducts. Also, parasitic worms can burrow into the skin. If you're walking on a beach and you step on a parasitic worm, and it can burrow up in through the bottom of your foot and enter into your body that way. It can get in through a bite or a scratch. Somehow the skin, although it's a great barrier, can be broken. And we do have these hair follicles and sweat gland ducts where microbes can work their way in. There's also a parenteral route of entry. Those are puncture wounds that can introduce the bacteria. That would be like through an injection, or if you were stabbed, or you had a very deep cut. Somehow the skin is broken. You could be having a surgery and the bacteria enters through a surgical incision. These are parenteral routes. The ID50 is the infectious dose of pathogenic cells, virions, or toxins required to cause infection in 50% of the population. The LD50 is the lethal dose of pathogenic cells, virions, or toxins required to kill 50% of the population. And we're going to do a review question later on in this lecture, and it will help you with understanding ID50 and LD50. For now, you need to know that ID50 is the infectious dose required to infect 50% of the population, and lethal dose, LD50, to kill 50% of the population. Remember, bacteria have different structures that can be virulence factors for that bacteria. In the case of adhesion, we've seen fimbriae and pili. We've seen capsules and slime layers that help them to stick to mucous membranes. And we know that biofilms can attach to surfaces and are very difficult to remove with disinfectants. That's a virulence factor of that bacteria. Adhesion is a virulence factor. They also can produce enzymes, and these enzymes can do different things like coagulate blood or destroy clots in blood and make you bleed out. We will talk about a few of these on the next slide. Remember, we've talked about toxins, endotoxins and exotoxins. Gram-negative bacteria have endotoxins in that lipid A portion of the LPS layer. Most exotoxins come from gram-positive bacteria. Bacteria also can evade the immune system. They can evade phagocytosis. The capsule is a very important structure that helps them, in addition to being able to stick to things like mucous membranes, but evade the immune system. Fimbriae also can do that. And remember, in Mycobacterium, they have that mycolic acid. That mycolic acid also helps to protect the bacteria from the immune system, helps them to evade phagocytosis. Bacteria also can do what's called antigenic variation. They can alter their surface proteins in order to disguise itself from the immune system. So maybe the immune system recognizes this foreign thing that's entered into its body and it gets ready to attack it. And then when it goes to attack it, that bacteria has changed its surface proteins, and now the immune system can't recognize it, and so it's able to evade the immune system. Again, bacteria can produce enzymes that are a virulence factor for that bacteria. They can have coagulase, which coagulates blood, or kinases that destroys blood clots and makes you bleed internally. They have many different types of enzymes, and they use these enzymes for a particular thing, to destroy antibodies or to suck up all the iron. There's many different things that they can do to cause disease and to increase the extent of disease because of these virulence factors. Toxins, again, are those endotoxins and exotoxins. Toxins are also substances that contribute to pathogenicity. Toxigenicity is the ability to produce toxins to cause damage to the host cells. Toxemia is a definition that you really need to know, and it means the presence of the toxin in a host's blood. Toxoid is an inactivated toxin, and that's used in different types of vaccines, like a tetanus vaccine. An antitoxin is antibodies that can be used against a specific toxin, like in a treatment. We are going to talk a lot about endotoxins and exotoxins, so let's revisit them again. It's very important to understand in microbiology, especially when we're talking about infections and illnesses and how they are treated and why we treat them certain ways. So again, an endotoxin is found on gram-negative bacteria. It is part of that LPS layer of gram-negative bacteria. It is that lipid A portion. The endotoxin is liberated when the bacteria die. When that bacteria dies, it releases that endotoxin. What happens if someone is infected with a gram-negative bacteria and they receive an antibiotic that kills that gram-negative bacteria? It can release its endotoxin, which can cause very serious signs and symptoms to occur in the patient. It can even lead to sepsis, and it can become fatal. Endotoxins are very serious, and when you treat a gram-negative bacteria, you have to treat it in a certain way so that you don't release that endotoxin to the body, which can destroy the cells of your body in many different ways. So an exotoxin is produced mainly by gram-positive bacteria. Gram-positive bacteria mainly produce exotoxins, and these exotoxins can target the specific receptors on specific cells and damage those specific cells, but not all cells, like specific ones that it wants to target. An exotoxin can either be secreted by a bacteria or it can be released by a bacteria when it is lysed, like in a gram-negative bacteria being lysed and releasing its endotoxin. The same thing can happen for an exotoxin, but exotoxins are gram-positive bacteria mainly, and endotoxins come from gram-negative bacteria. Portals of exit are pretty much the same as portals of entry for most microorganisms, just in a little bit of a different manner. For instance, like the respiratory tract, instead of inhaling them as a portal of entry, you exhale them or cough or sneeze them out. That would be a portal of exit for that virus or for that bacteria. You can have the portal of exit in your feces for the GI tract, the digestive tract, or it can also come out of your saliva, out of your spit. A portal of exit can be your urine, or it can be in your genital secretions. Your skin sweats. Microorganisms can be released through that sweat. Another very important portal of exit that you need to understand is blood. So blood from a puncture wound, blood from a cut, blood from a head injury, however the blood is released from the body can be a portal of exit for microorganisms to be transferred to someone else. Biting arthropods. For instance, the mosquito and malaria. If someone has malaria and a mosquito comes along and bites that person, when it is sucking that blood up, it is introducing that protozoa into its body and it can transfer that to another person. So blood is a very important portal of exit that you should understand. Let's do a few clicker questions. The virulence of the common cold compared to influenza is A, lower, B, about the same, C, higher. The answer is A. It's lower. The virulence, remember, is the extent of harm. What is the degree of that infection? How serious is it? The virulence of the common cold is usually much lower than influenza. The flu can make you very, very sick, and it can be fatal. So the virulence, the extent of harm that can be caused, the virulence of the common cold is A, lower compared to influenza. You feel tired and are starting to have a slight sore throat. It's a little bit scratchy, and your nose is starting to be runny. This is which period of disease? Is it A, incubation, B, prodromal, C, disease, D, convalescent? The answer is B. It's the prodromal period. It's where you're just starting with mild signs and symptoms. You feel a little bit sick. You're just starting to get sick. Remember, the disease period is where your signs and symptoms are very obvious and you're very sick. In that incubation period, remember, those pathogens are multiplying. They have just been introduced to your body, but you do not have signs and symptoms yet. So if we think about COVID-19, you can have COVID-19, be infected with COVID-19 for 2 to 14 days before you are showing signs and symptoms. That's still the incubation period. You are transferring this infection to other people, and you may not know that you have it. It is not a prodromal period. You don't have signs and symptoms. So think of the periods and kind of relate them to infections. So again, for this question, you feel tired, starting to have a slight sore throat and a runny nose. This is the prodromal period. Which specimen has the highest level of pathogenicity? One with an ID 50 of 2,000, an ID 50 of 20,000, an LD 50 of 2,000, or an LD 50 of 20,000. Remember, ID 50 means it is the infectious dose of something that causes 50% of the people to become sick, to become ill, to become infected. LD 50 is the lethal dose that causes 50% of the people who are infected with this particular bacteria or virus or microbe to die. So if you look at A for the infective dose 50, the ID 50 of 2,000, it takes 2,000 particles of something to make 50% of the population sick. If you look at ID 50 of 20,000, that means it takes 20,000 particles to infect 50% of the population. Now let's look at lethal dose. Lethal dose 50, LD 50. In C, it takes 2,000 particles to kill 50% of the population. For D, LD 50 of 20,000 means it takes 20,000 particles in order to kill 50% of the population. So if you're looking for which specimen has the highest level of pathogenicity, the answer is C. Because you need to think about what is the worst thing? What is the highest level? The highest level would be death. That's the worst thing. Death is far more serious than an infection. So LD 50 is far more serious than an ID 50 of something with the same number of particles. So first, you need to look at the LD 50 then. So then you would pick the lowest number because it takes the least amount of something to kill someone. So the answer to this is C. The LD 50 of 2,000, only 2,000 particles to kill 50% of the population. That is the highest level of pathogenicity. E. coli is a gram-negative bacteria that produces a toxin. This toxin is A. Endotoxin, B. Exotoxin. The answer is A. Gram-negative bacteria have endotoxins. So E. coli, which is a gram-negative bacteria, has an endotoxin. Staph aureus is a gram-positive bacteria that produces a toxin. This toxin is A. Endotoxin, B. Exotoxin. The answer is B. Exotoxin. Staph aureus, which is gram-positive, is producing an exotoxin. Remember, most exotoxins come from gram-positive bacteria. For respiratory infections, the portal of exit is coughing and or sneezing. Is that A, true, or B, false? The answer is A, true. And that's the end of chapter 15. Very short, but full of great information.